Welcome to Public Health on Call, a podcast from the Johns Hopkins Bloomberg School of Public Health, where we bring evidence, experience, and perspective to make sense of today's leading health challenges. If you have questions or ideas for us, please send an email to publichealthquestion@jhu.edu. That's publichealthquestion@jhu.edu for future podcast episodes.
This is Lindsay Smith-Rogers. Today, Stephanie Desmon talks to George Dimopoulos, a mosquito expert at the Johns Hopkins Bloomberg School of Public Health, about his new findings that could someday help prevent the spread of mosquito-borne diseases like dengue, Zika, and chikungunya. They discuss the quirks of the mosquito's immune system that allows them to spread viruses but not succumb to them, and how researchers might be able to capitalize on that to prevent the spread of disease. Let's listen. George Dimopoulos, thanks so much for joining me.
Thank you, Stephanie.
You're here today because I want to ask you about a new discovery that you and your colleagues made that has to do with the Aedes aegypti mosquito. And in English, that's basically the mosquito that transmits Zika and chikungunya and dengue and all kinds of human diseases. I'm curious if you could tell us a little bit about what you found.
Yes, we have been studying mosquito infections with these human pathogenic viruses. And one interesting observation is that these mosquitoes don't seem to get sick from infection. And that's why they can fly around and they can transmit these viruses. It turns out that the mosquito has an immune system like humans and other organisms. And this immune system is also fighting these viruses. And there is an antiviral defense system in the Aedes mosquito called the RNA interference system that is fighting these viruses. What we found in our study was that if we inactivate a key protein of that defense system called Argonaute 2, the mosquitoes become very susceptible to infection, so they get highly infected. But more importantly, the mosquitoes also get sick from infection and they die from infection. So this is very interesting to us because we basically have created a mosquito now that cannot sustain infection and thereby cannot transmit infection. It turns out that this Argonaute 2 protein is involved in defending the mosquito against infection, but it's also involved in helping the mosquito to tolerate infection. And that's why the wild type mosquitoes that have an Argonaute 2 can survive infection. So when we remove Argonaute 2 from the mosquito using genome editing tools, these mosquitoes will die from infection because the mosquito cannot remove the damaged cells. Infection will cause cell damage in different tissues of the mosquito. And if those damaged cells are not removed, they become toxic to the mosquito and the mosquito dies. So when you inactivate Argonaute 2 from the mosquito, then these mosquitoes will sustain high infections, a lot of tissue and cell damage that cannot be repaired or removed, and they will die because of that.
So how would you go about, obviously you're doing this in your lab. Is this something that could be used in the wild? And if it was, would it kill off all the mosquitoes?
That's a very good question, Stephanie. And this is something that we find very interesting with regards to the Argonaute 2 protein and the possibility of engineering mosquitoes in a way that you would inactivate Argonaute 2 when they get infected with these viruses, because that would result in the death of the mosquito and the mosquito would not be able to transmit the virus. The beauty of this approach, if you can get it to work, is that you would only kill mosquitoes that get infected with the viruses, right? So, and the majority of mosquitoes in nature, they will never get infected with any of these viruses because these infections are not that common. So even in a highly dengue endemic area, the great majority of mosquitoes would not end up becoming infected because they would feed on uninfected humans. And the mosquito is only going to feed maybe 3 or 4 times on human blood throughout its lifespan. in theory, we could develop a mosquito that would only die when it feeds on an infected human. So that would not really cause a change in mosquito prevalence or have any ecological impact.
Obviously, that's a concern whenever you're introducing something new or want to introduce something new. Talk a little bit about that, please.
Yes, it is a concern to, for example, start to kill off all the mosquitoes. However, Aedes mosquitoes is only one species of many different species of mosquitoes out there, and removing one species of mosquitoes is probably not going to have any impact on ecology. It also turns out that mosquitoes are not playing a very important role in the food chain. So there are many other insects out there that various predators can feed on. And there have been actually quite a few discussions on this topic, on whether killing off a very large number of mosquitoes of a certain species would have any impact on the ecology. And the opinion is that it would most probably not have any impact. Another proof of that is the relatively recent introduction of a closely related species to Aedes aegypti, which is called Aedes albopictus, into the Americas. So this mosquito species did not exist in the United States 20 years ago and has been introduced recently. And now it's found throughout the country. And this is an invasive species. So if you would remove it, you would obviously just go back to the situation where we were when it didn't exist. So most likely removing even an entire species of mosquito is not going to have any ecological impact. And there are hundreds and hundreds of different mosquito species, plenty of mosquitoes for predators to feed on, even if one would be removed.
It seems like such an enormous project. Like, how would you get these mosquitoes circulating?
Well, this is something that can be solved through so-called gene drive technology, which is a engineered genetic technology by which you can spread a gene or a mutation into a population of mosquitoes in a way that it would not obey the classic Mendelian laws of inheritance. So basically what happens when you use a gene drive technology, when a genetically modified mosquito mates with a wild type mosquito, all the offspring become genetically modified. So very quickly, you can introduce that gene or that mutation in a population through just a few generations.
I can imagine people out there concerned about crazy sci-fi effects that we hadn't considered ahead of time. Is that something you all think about?
Most definitely, and that's why this technology has not been used yet. In nature, even though it has existed in the labs for many years, there is a lot of testing going on, a lot of discussions of possible things that could go wrong. There has also been a significant effort in developing mitigation technologies. So you can use these CRISPR-Cas9 gene drive technologies to spread mutations in a population, but you can also use the same technology to revert a mutation into the wild type state again. And there has been a lot of discussions with the scientists from different areas, the possible things that could go wrong. And it seems like this technology does not really pose any real danger, especially with regards to, mosquitoes. And most likely the gene drive is going to break down because of mutation on its own. So one would have to apply it multiple times sequentially. It's very unlikely that a gene drive introduced mutation is going to spread to all the mosquitoes of that species on the planet.
So you also study malaria, and I'm curious if this offers any hope for similar solutions to malaria.
Argonaute 2 and the RNA interference pathway are strictly antiviral defense systems, and the malaria parasite is not a virus, so we would not be able to use this particular Argonaute 2 factor to develop a malaria control strategy. However, similar concepts for controlling malaria and malaria-transmitting mosquitoes have been developed. And here we are looking at ways by which we can engineer the Anopheles mosquitoes that transmit malaria so that they would also die when they get infected with a malaria parasite. Because similarly to virus infection in Aedes aegypti, also the malaria parasite in the Anopheles mosquito is causing some damage to the mosquito through its infection. And one could find ways to make that damage lethal to the mosquito. So we are looking into that possibility.
So I guess, how likely are we to see this anytime soon? I mean, I know this is something you just discovered in the lab. Do you see, obviously you'll need further testing?
I think we will see GM and gene drive-based technologies for malaria control relatively soon, not based on this particular pathway. For Aedes aegypti, it's going to take a bit longer because we are not as advanced in our research on these technologies with Aedes aegypti. It's going to take several years to see these technologies actually reaching the implementation stage. And maybe that sounds discouraging, but it's important to remember here that these diseases have been with mankind forever. Malaria is an ancient disease, and it has been killing millions and millions of people for thousands of years. So whether something is going to take another 5 years or 10 years, it's going to be very important to actually get it to work, regardless of how long it's going to take. Usually the quick fixes, they don't last very long. And we have learned that lesson from malaria. We have had drugs for over 100 years. We have had insecticides for almost 100 years. And yet there's a lot of malaria. And the same thing applies for the Aedes transmitted diseases. So sometimes it's better to spend some time and come up perhaps with a new method, a new technology that could have a much greater impact compared to the technologies used until now.
So I guess that I'm still going to get bitten for a long time in my backyard, but hopefully down the road, the mosquitoes will not make me sick.
Well, Stephanie, assuming that you're in Maryland, I don't think you will have a problem, even if you get bitten a lot. But if you did have a place in a dengue endemic area, then I would suggest you to use some methods of suppressing the mosquito populations, maybe spraying some insecticides or using some of the larvicidal methods that exist for Aedes aegypti.
So climate change is changing where we can find these mosquitoes. Do you think we're going to see more of them this far north?
It's hard to tell because climate change can have two effects on mosquitoes. It can reduce mosquitoes because it might get too warm for the mosquitoes or too dry. It can also introduce mosquitoes to new areas. So it is possible that we will see more of the Aedes mosquitoes, and especially the Aedes aegypti mosquito in the United States. As climates get warmer, we have longer summers and warmer winters. It is possible to make that prediction, although I am not sure whether such trends have been observed yet.
George Dimopoulos, thanks so much for joining me.
Thank you, Stephanie.
Public Health on Call is a podcast from the Johns Hopkins Bloomberg School of Public Health, produced by Joshua Sharfstein, Lindsay Smith-Rogers, and Stephanie Desmon. Audio production by J.B. Arbogast, Holly Cardinale, Philip Porter, Spencer Greer, and Matthew Martin, with support from Chip Hickey. Distribution by Nick Moran. Production support from Catherine Ricardo. Social media run by Grace Fernandez and Shian Briscoe. If you have questions or ideas for us, please send an email to publichealthquestion@jhu.edu. That's publichealthquestion@jhu.edu for future podcast episodes. Thank you for listening.